Good morning, everybody. Thank you for giving me the opportunity to present some of the work we have done in West Bengal Forest Department. I am 1992 Indian Forest Service officer from West Bengal cadre. I will focus my presentation on the tree medicinal species of tree habit, especially that we have carried out in West Bengal. The herbs and the shrubs are also important, we agree, but in Forest Department we are basically focusing on the tree species. Our working plan prescriptions as such in West Bengal, they do not exactly specifically bring out any particular focus on tree medicinal species, but it is just mentioned like sal and associates or endemic species to be planted in various areas in West Bengal, but we would like the specific prescriptions to be incorporated during the next revision of the different working plan prescriptions in West Bengal, so that I'll come to that later. The priority conservation species are incorporated within the working plan itself. As in other areas, we have got in situ conservation areas as well as ex situ conservation areas. We have recently concluded FRDHT associated UNDP CCF2 project in West Bengal, under which we demarcated 7 MPCAs. In West Bengal, we have got 4 biogeographical zones. So these 7 MPCAs covered all the 4 biogeographical zones and belong to 4 forest types. In West Bengal, we have got 10 forest types because we have got a very wide distribution from the Himalayas down to the mangroves. But within our MPCAs, we covered 4 forest types. These areas were under the jurisdiction of 6 different forest protection committees under our JFM systems. We had a camp workshop in 2007 in West Bengal, which identified 46 species. Just a moment. Within the 46 species, this is the habit distribution, like 15 were tree habit. So we were subsequently focusing on those 15 species. Within the 7 MPCs in West Bengal, we had 2 in southern part and the rest were in northern Bengal. The first 3 on this list, the first one is within the Buxar Tiger Reserve. We inventorized sample plots within the MPCAs, and within the sample plots we found, out of the 46 species of conservation priority for West Bengal, we found 10 of the tree species within those sample sites. It is possible that several more are within, but we did only sample inventarization within the project. We are going to cover more areas subsequently. The project has just ended in June, but we found in the northern Bengal, the first 3 belong to the North Bengal plains, the Terai Duars areas. They have the most priority species within the MPC areas. The last 2, Tonglu and Dhotre, these are the Darjeeling areas. So within that, these are the different areas, but with Dhotre has the flagship species of Taxus. Apart from Taxus, there are other important herbal species like Chiraeta and Panax Sindoginseng within those MPC areas, which we are trying to protect the natural populations. In Tonglu area of the Darjeeling, there is Berberis aristata, but there is also within that area we are getting other herbal, important herbal populations of 3 species of Aconitum. So these species, these are totally hands-off areas. In future, we are hoping to propagate these areas within our nurseries for distribution elsewhere. In Sundarban areas, we have got one, that is the Bonny Camp, Nipa fruticans, but we haven't done much work there. We have just inventorized the species. In the southern Bengal plains, there are other species, but for us, we have mainly worked on the taxus that I'll detail later. Okay, the camp identified 15 species where these are the 15 species with the IUCN vulnerability category. Apart from the camp and FRLHT funded project, we under the National Medicinal Plant Board, we have demarcated and we are still protecting 41 hectares of different gene pool areas within parts of West Bengal. Apart from these in situ areas, we are carrying out plantations of medicinal trees with funds from different sources like Medicinal Plants Board, and within from our state plant budget, from different CSC schemes. We are planting specifically these species. There are several more also. These are not in pure block plantations. These are usually mixed plantations. This is creating some other problem where it is good to have mixed plantations, but then we are not getting any seed stands. We are getting individually scattered seed trees within the forest, so it is becoming difficult for us to predict, like within 10 years when the seeds start coming, what is our potential production. So these are all mixed forests we are bringing up. We are trying to stress on supply of quality planting materials. So we have got plus trees and then we have got seed trees, because we are not getting pure block plantations. So it is like scattered seed trees in the forest, which we are tagging and keeping track of. So different trees we have on our records, the different seed stands, species wise, area wise, which we are adding to every year. But we are seeing that seed stands are comparatively lower, more we are getting seed trees. I'll focus on a few species which we are working on, which it is of local importance to us. Now, Taxus vallecina is the same as Taxus baccata. We have been planting in West Bengal since 1994. It is a very slow growing species. Since 2002, we have got a series of plantations. It is a very small patches. 2002 plantation currently is around this high only. I mean, the plant survival is full, but it is like this high only from 2002. 1994 plantation, this picture is in 1994 plantation. It is comparatively taller. We are raising seedlings from 2 plus trees. We have got cuttings, etc. Recently, I sent a sample from the 1994 plantation for phytochemical testing. Quantitative testing could not be done, but qualitatively they have detected taxol and taxine in the leaves, the young shoots, and in the bark. The largest amount has been reported in the leaves, but we'll have to go in for quantitative testing. We are wanting to go in for age gradation, location specific, etc. Because the earlier publications in West Bengal were from high forest samples. So these will be from our plantations. So we are going to spread it over and see, because the high forests, we are not going to allow a collection from our high forests. But we can raise the plantations from which, and if we can find comparable phytochemicals in our plantations as with the high forest, then we can say that kindly take the samples from the plantations and leave the high forests alone. There's also Nageshwar. This is Mesoaferia. We have got seed stands for this. We have a 100-hectare preservation plot with almost 75% composition of Mesoaferia as a pure patch. We are using this species in our plantation since 2004. Again, these are not pure block plantations. We have not raised pure block plantations, so it is scattered, and but they are doing very well. Cinnamomum sesidodaphne, this is an endangered plant also. We are again including these in our mixed block plantations of the medicinal trees in West Bengal. We have several plus trees for this, from which we are collecting the QPM. Maculus villosa, this is a critically endangered plant because the bark is stripped and then the plants get endangered. And we are raising plantations, again not pure block plantations. Main constraints, like I said, is lack of seed stands. We have got scattered seed trees, but that is not adequate. Currently, the silviculture divisions, I am the conservator forest and research circle, so I've got 3 silviculture divisions covering entire West Bengal under my jurisdiction. We are raising around 25 hectares per year of this kind of plantations. Even if we wanted, we cannot step up our plantation target because there is not adequate seed supply of quality standards. And there might be seeds, but we are not sure whether the phytochemical content, etc., will be of standard, which is going to be of importance of medicinal value. As such, the tree species are slowly this thing, and trained personnel within the forest department, because we are using hycopods, etc., but for any particular studies that we have to take the measurements, etc., our field staff are so much engaged in with the other forest directorate works. This has to be like in collaboration with some research institute. We do not yet have a forest research institute in West Bengal, though it is on the anvil. Hopefully it will come up within the next 2 years. So it is normally in collaboration with the universities or other technical university institutes within West Bengal, that they take our samples, give us the results, and things like that. There's a lack of awareness within West Bengal regarding the source of like Machilas bark. They are using it for agarbatti, but we don't know. And there's a lot of social political disturbance, especially with the Maoist infestation in West Bengal. So it is making the afforestation activities very slow. Future trust areas are creation of more seed stands, incorporation within the working plan. We are wanting in-house facilities. If the State Forest Research Institute gets created, then this hopefully will take care of this. With identification of superior strains, then we can supply quality planting material for the other divisions and even to other states if they wanted. And we want to augment our knowledge base about the uses and the different occurrence of the different phytochemicals of importance. I deal with the silviculture and the research. Within the Forest Directorate, the marketing is dealt by a separate division. My colleague from the division is here. He will be addressing us in the session 5 today, so I left the marketing part to him. So any questions?